Welcome back to Dr. Liu's VCE Biology. Today's topic is changes in species over time, as presented in the 2024 Victorian Certificate of Education, VCE, Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. This diagram visualizes how different fish groups have evolved and diversified over geological time. The wider the shaded area, the greater the diversity of that group. Interestingly, the total diversity of fish 201 million years ago was less than what we see today. Most of the fish fossils discovered are about 300 million years old, not 359 million. We also learn that the very first fish group appeared over 485 million years ago, and spiny sharks died out 298 million years ago. A. Incorrect. Carbon-14 dating is only effective for much younger fossils, under 50,000 years, making it unsuitable for spiny shark fossils that are hundreds of millions of years old. B. Incorrect. While transitional fossils show evolutionary links, they don't directly tell us the relative age of individual fossils. Their value lies in demonstrating evolutionary pathways, not in dating. C. Correct. Faunal, fossil, succession and stratigraphy are the appropriate methods. By examining the rock layers and the specific fossils found within them, we can accurately determine the relative age of the spiny shark fossils. D. Incorrect. Comparing fossils would be relative dating, not absolute. Furthermore, muscle tissue would not be preserved in fossils of this age, making that part of the option fundamentally flawed. This diagram illustrates the evolution of dolphins, showing changes from their earliest ancestors to their most recent forms. A key observation is the gradual reduction in the prominence of the pelvis structure over geological time. This diminishing feature provides valuable insight into evolutionary history, as structures that were once functional in ancestral species can become reduced in size or complexity. Consequently, the dolphin pelvis bones, having lost their original function through evolution, are considered vestigial structures. Researchers sampled 20 populations of E. salubris, observing notable differences in tree appearance and leaf thickness. These trees fell into two distinct groups, those with thicker leaves, image A, and those with thinner leaves, image B. While these groups live in close geographical proximity, the thinner-leaved trees are exclusively found in soils with high phosphorus levels. Further genetic analysis revealed significant differences between the two groups. These compelling findings strongly suggest that these two E. salubris groups should be classified as separate species. Fieldwork refers to the actual process of going out into the natural environment where E. salubris trees grow. During this process, scientists classify the data collected and identify the species or distinct groups within it. Therefore, the scientific methodology employed to collect the E. salubris samples inherently involved fieldwork, which led to the classification and identification of the two distinct groups. The evidence that suggests the two groups of E. salubris are separate species includes different structural morphologies. They noted differences in the appearance of the trees and the thickness of the leaves. Different ecological niches or growth conditions. The two groups, despite close geographical proximity, exhibit a clear preference, with thinner-leaved trees only found in soils with high phosphorus levels. Significant molecular or genetic differences. Genetic analysis comparing the two groups showed significant differences, implying different DNA or amino acid sequences to molecular homologies. The two groups of E. salubris are closely related species living in the same geographic area. Therefore, Their evolution is considered an example of sympatric speciation. The genetic diversity observed between the two groups of E. salubris likely arose from a combination of the following factors. Initial variation. There was likely pre-existing natural variation in alleles and phenotypes within the ancestral E. salubris population. Differential selection pressures. The two groups were exposed to different environmental challenges, such as varying levels of phosphorus in the soil. Selective advantage. The thinner-leaved trees likely had a selective advantage that favored their survival and reproduction, specifically in soils with high phosphorus levels. Reduced gene flow. Over time, the exchange of genetic material between the two diverging groups became limited, leading to reduced gene flow. Accumulation of mutations. Separate, independent mutations accumulated within each group, further increasing their genetic divergence. Reproductive isolation. Factors such as differences in flowering time or pollination mechanisms may have led to reproductive isolation, preventing interbreeding. It's crucial to address potential misconceptions regarding genetic changes within a population over time. For instance, some students incorrectly apply Lamarckian ideas, suggesting that individual trees adapted to high phosphorus levels in the soil through an active process during their lifetime and then passed these acquired traits on. Instead, genetic diversity arises from random mutations and selection acting on pre-existing variation. Also, many students tend to provide a generic speciation response when asked about genetic diversity between diverging groups. It's important to clarify that this question focuses on the mechanisms that led to the genetic differences between two already separating populations, rather than merely stating the outcome of speciation, e.g., the inability to breed and produce fertile offspring, which isn't directly the cause of the genetic diversity itself. This section is worth 11 marks, constituting 9.2% of the total assessment, 120 marks, and is divided into two question types: multiple choice questions, 3 marks. Question 31, Fish evolution, diagram analysis of fish group evolution and diversity over geological time. Question 32, fossil dating, understanding absolute, carbon-14, and relative, superposition, dating methods. Question 34, dolphin evolution, identifying vestigial structures, pelvis bones, in dolphin evolution. Short answer question, 8 marks. Question 8a and 8b, Eucalyptus Salubris Case Study, analyze research on E. salubris in Western Australia, focusing on evidence, morphology, ecology, genetics, for 2 distinct species. Question 8c1 and 8c2, speciation and genetic diversity, explain sympatric speciation and list factors contributing to genetic diversity within the E. salubris groups, e.g., variation, selection, gene flow, mutations, reproductive isolation. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.